In human and animal reproduction, the math is usually simple. One sperm fuses with one egg, but flowering plants operate on an entirely different system. When a pollen tube arrives at an ovule, it delivers a 2-for-1 special, 2 distinct male gametes, each with a highly specific job. To get those gametes inside, the pollen tube needs directions. It follows a chemical trail secreted by the synergid cells, specifically through a structure called the filiform apparatus, which guides the tube directly into the ovule. Look at this diagram of a mature embryo sac. Down at the micropilar end, we have the haploid egg cell. Then, taking up the large central cell, we find 2 haploid polar nuclei sitting together. If we fade out the supporting structures, the antipodals and synergids, we isolate the exact targets. One gamete will hit the egg, and the other will hit the polar nuclei. This dual targeting allows the plant to simultaneously build a new embryo and a dedicated food supply to keep it alive. This diagram shows the sequence unfold. At the micropyle opening, the pollen tube bursts, releasing 2 haploid male gametes. Event 1 is syngamy. The first gamete fuses with the haploid egg cell, creating a standard diploid zygote. That zygote is the future plant. As a historical note, this specific fusion was first observed by Edward Strasburger in 1884, studying this exact plant, the ghost plant, Monotropa. Event 2 is triple fusion. Moving back to our diagram, the second male gamete travels to the central cell, fusing with the 2 polar nuclei. Counting the nuclei, 1 haploid gamete plus 2 haploid polar nuclei creates a triploid, or 3N, primary endosperm nucleus. Syngamy creates the plant. Triple fusion creates the food. Together, happening in the exact same embryo sac, these 2 events constitute double fertilization, a mechanism unique to angiosperms, first explained by Nowoshin in 1898. This brings up a frequent board exam question. After syngamy, why does the newly formed zygote remain completely dormant for a period of time instead of dividing immediately? It comes down to resource management. The zygote waits for a guaranteed food supply, staying dormant while the tricloid primary endosperm nucleus rapidly divides into nutrient-rich endosperm. Only when the food is ready does the zygote wake up to form the embryo. Examiners love to test the math behind this process. Look at this previous year question. When a diploid female plant is crossed with a tetraploid male, what is the ploidy of the endosperm cells in the resulting seed? Let's break down the inputs. The female plant is diploid, or 2N. Her meiosis works normally, so her 2 polar nuclei inside the central cell are both standard haploids, N and N. The twist is the male plant. Because it is a tetraploid, or 4N, meiosis cuts that number in half. The male gamete arriving at the embryo sac is therefore diploid, or 2N. Apply the actual triple fusion equation, the 2 maternal polar nuclei plus the paternal gamete, in this case, N plus 2N. The resulting endosperm is 4N, or tetraploid. With fertilization complete, the microscopic work is done. Petals and sepals wither away, and the entire structure enters its final developmental phase. The internal components mature into functional units. The diploid zygote develops into the mature embryo, and the primary endosperm nucleus becomes the endosperm mass. Zooming out, the entire ovule hardens into the seed coat, while the surrounding ovary swells and ripens into the fruit. To secure top marks, you need this comparison mapped out clearly. Syngamy, LUT gamete and the egg cell yield a 2N zygote. Triple fusion, 1 gamete and 2 polar nuclei yield the pen. 2 parallel equations running at the exact same time. Nailing the mechanics of double fertilization prepares you for the toughest exam questions. More importantly, you now understand the exact biological sequence that produces the seeds and fruits, making up nearly all of the global agricultural food supply.